Hello, my dear students, and welcome back. Exams are just around the corner. I know, the pressure is building. But don't you worry, because today we are going to tackle one of the most important topics, genetic disorders, and we're going to make it super, super easy. Now, this chapter on genetic disorders, it might look a little scary in the textbook, but trust me, it's one of the most logical and high-scoring chapters you'll find. I'm here to break it all down for you, so you can just walk into that exam and ace this section. So are you ready? Let's get started. Okay, so first things first. Just imagine our body is built using a massive set of instruction manuals. Every single cell has a copy. These manuals, let's call them the books, are our chromosomes. And the actual instructions written inside those books, those are our genes. Simple, right? This is the complete blueprint for building you. Now, just like with any manual, sometimes there can be an error. It could be a really big one, like a whole book is extra, or maybe a book is missing. Or it could be a really small mistake, like a single spelling mistake, a tiny little typo in one of the instructions. And these errors, my dears, are what we call genetic disorders. So let's start with those big mix-ups. The problems where the total number of our chromosome books is wrong. The proper scientific term for this, and you should remember this for your exams, is a numerical chromosomal abnormality. The most common example, and it's an absolute favorite for exam questions, is Down syndrome. Now, if there is one thing, one number you have to remember for this, what is it? It's 21. Just lock the number 21 in your brain. Now, have a look at this. This is called a karyotype, which is basically a picture of all our chromosomes lined up. And if you look closely at chromosome number 21, you see it? There are 1, 2, 3 copies instead of the usual 2. That's why we call it trisomy 21. Dry just means 3. Simple. And this picture, this is exactly what you might see in a question paper, so pay attention. This happens because of a little error during cell division called non-disjunction, where the chromosomes just fail to separate properly. That's the secret behind that extra copy. So what are the symptoms you need to write about? They often have a flat face, eyes that slant upwards a bit, and a small mouth with a tongue that might stick out. They also tend to have a short neck and limbs, and look for this one, a single deep line or crease right across their palm. You just remember these few points, and you can easily score full marks on this question. Excellent, you are doing so well. Now, what if the mistake isn't in chromosome 21, but in the sex chromosomes? You know, the X and Y chromosomes. Well, this leads us to 2 other very critical syndromes you absolutely need to know. And this slide is just brilliant for a quick revision, so go on, take a screenshot. Klinefelter syndrome affects males. They have an extra X chromosome, which makes their genetic blueprint XXY. On the other hand, Turner syndrome affects females. They are missing an X chromosome, so their blueprint is just X, or sometimes written as XO. A simple way to remember, Klinefelter's has something extra, Turner's has something missing. All right, so we've covered the big mix-ups with the chromosome books. Now let's zoom in way in, and look at those tiny little typos, the errors that happen in just a single gene. A monogenic disorder is exactly what it sounds like, a condition caused by a mutation in a single, or mono, gene. And the textbook example, the one we really need to understand, is sickle cell anemia. Just look at this picture. It tells the whole story, doesn't it? On the left, you see these nice, round, healthy red blood cells, perfect for carrying oxygen. But on the right, just because of one tiny typo in a single gene, the cells get twisted into this crescent or sickle shape. These sickled cells can't carry oxygen properly, and they get stuck, blocking blood flow. The difference is just so clear. Okay, now for this very important concept, autosomal recessive. Let's break it down. Recessive means you need to inherit 2 copies of the defective gene to actually get the disease, one from your mother and one from your father. If you get only 1 copy, you are what we call a carrier, but you won't have the full disease yourself. So the rule is simple. 2 bad copies cause the disease. 1 bad copy makes you a carrier. So how do we track these genetic traits through a family? Well, we get to play detective. We use a special tool called Pedigree Analysis. It's basically a fancy family tree that lets us see how a trait is passed down from one generation to the next. It's a really powerful way to track these disorders. And these are our detective symbols. The rules are super simple. A square is for a male, a circle is for a female, and if the shape is shaded in, it means that person is affected by the trait we're studying. A horizontal line connecting a square and a circle, that means they're parents. It's really just a logical puzzle with its own set of rules.
All right, detective hats on, it's your turn. Here's a quick question. If both parents are carriers for sickle cell anemia, meaning their genotype is big S, small s, what is the probability, what is the chance that their child will actually have the disease? Take a moment to think about it.
And the answer is a 25% chance. That's 1 in 4. If you draw out that little Punnett square with big S, small s from both parents, you'll see that one of the 4 possible boxes is small s, small s. This is a perfect throwback to Mendel's laws, which you've already learned. You see, everything in biology is connected. We are almost at the end now, and I have created a very special slide for all of you. Think of this as your ultimate summary. It's perfect for a quick screenshot for that last-minute revision just before you walk into the exam hall. Okay, let's do a rapid-fire round. Down syndrome, it's chromosomal, specifically trisomy 21. Klinefelter syndrome, also chromosomal, it's 47XXX in males. Turner syndrome, chromosomal again, 45X in females. And finally, sickle cell anemia. That's a monogenic one, and it's autosomal recessive. This one table has all the keywords you need to score full marks. And finally, I just want to say this to all of you. You are brilliant and you are so well prepared. Just revise this one more time, go over your notes, and you are going to do wonderfully in your exam. I have complete faith in you. All the very best.